we'll sort it out from there. The order of presenters is going to be based off of what is at the bottom of the screen here. So we'll start off with, we'll go with Claire then, it looks like, and then Katie will go with later. So I'm going to go ahead and add you to the screen, Claire, just so you know.
Hello.
Hello. So I'll let you go ahead and share. So just so everybody knows how this is going to run, so these are going to be basically 10 minutes, right? So you have about 8 minutes for your presentation. I'm going to, once you start, close down my video so that way you're not seeing me. You'll go for about 6 minutes, and then I'll just add my video back without my face. So that way you know it's kind of a 2-minute warning, and I'll also just add a little bit of a note in the chat for you so that way you can see that. But and then once you have wrapped up, we'll answer any questions, and then we'll move on to the next speaker, okay?
Okay, that sounds good.
Awesome. And so if you have any questions as we go along, also feel free to post those in the chat, okay?
Can you see my presentation?
Yeah, we can see it.
Okay. So hi everyone. For my Polygence project, I did a review paper on the associations between childhood abuse and emotion regulation. So a little bit about me. My name is Claire Lim. I'm from New Jersey. I am currently a senior in high school and I'm homeschooled. And I'm very interested in psychology, mainly child psychology. And some things I enjoy doing in my free time are cooking and gardening. Before I started my Polygence project, I actually had no prior knowledge on child psychology or psychology in general. It became a fairly recent interest of mine. So while working on my project, I aim to get a better understanding of child psychology. So the reason why I decided to focus on child abuse for my project lies in the number of children that are abused around the world. According to the World Health Organization, 1 in 4 adults encounter physical abuse when they were a child. In just the European region, 18 million people have been physically abused, 44 million people have been sexually abused, and 55 million people have been emotionally abused when they were younger. It's even more alarming that 90% of all abuse goes undetected in the European region. And looking at this with a more global perspective, 1 out of 2 children, or approximately 1 billion children, encounter some form of violence each year. So in my review paper, the 3 types of child abuse I examined are physical, sexual, and emotional abuse. Physical abuse is determined when an adult intentionally hurts a child physically. Sexual abuse is determined when an adult engages in sexual activity with the child that the child does not fully understand and cannot give consent to. Emotional abuse is determined when an adult fails to provide a child with a supportive environment that allows them to develop emotional and social skills. So what exactly is emotion regulation? There are many different definitions of emotion regulation, but in my review paper, emotion regulation refers to the ability to manage one's emotions. So someone with emotion dysregulation would find it hard to manage their own emotions. Emotion dysregulation can manifest as excessive crying in children. It can also manifest as extreme outbursts of anger in both children and adults. For example, in a situation where an adult with emotion dysregulation has to wait longer than expected, the adult might react to the situation by getting angry, yelling, and throwing things. Emotion regulation is considered to be one component of self-regulation, which more broadly encapsulates behavioral, cognitive, and emotional components. Emotion regulation typically starts to develop at a very young age, similar to other components of self-regulation. So why is emotion regulation so important? Well, some signs of emotion dysregulation are depression, anxiety, and eating disorders. Although depression is a sign of emotion dysregulation can also serve as a predictor of depression. So people with emotion dysregulation may avoid situations that are difficult, use social media excessively, or abuse alcohol or other substances like drugs. Emotion dysregulation was found to be a contributor of physically aggressive and disruptive behavior in children. Some disorders that are associated with emotion dysregulation are ADHD, bipolar disorder, and borderline personality disorder. It can be very difficult for people with emotion dysregulation to lead healthy lives. These people can find it hard to maintain relationships due to increased emotion dysregulation. Children with lower emotion regulation were later found to have higher peer rejection due to increased expression of symptoms. It was found that children who experience physical, sexual, or emotional abuse are more likely to show concurrent emotion dysregulation problems compared to children who did not experience any abuse at all. This has been found to be true in preschool-age children and children between the ages of 6 to 12. The experience of abuse may lead to emotional pressures that can manifest as emotion dysregulation around the time children experience abuse. Not only that, children may have difficulties processing the experience of abuse, especially without the support of adults in their lives, assuming that these adults are abuse perpetrators. Children between ages 5 and 18 were found to have poor emotion regulation, increased avoidance, and increased emotional suppression and expression due to the abuse they faced earlier in their lives. A study on female college students found that women who encountered physical, sexual, or emotional abuse faced greater emotion regulation difficulties. In male college students, there was a connection between impulse control difficulties and emotion dysregulation. It was also found that women who remembered more childhood emotional abuse from their mother showed higher levels of emotional difficulties even a decade later. Some mothers who faced maltreatment in childhood were found to maltreat their own children due to emotion dysregulation. It is important to note that some of these mothers did not actually intend to hurt their own child. They found it difficult to manage their own emotions. This is possibly a good area of intervention because the mothers would probably be more willing to accept help from other people. Children who encountered abuse were found to have lower regulatory abilities if a parent was found to exhibit more negative emotions. Even if the child did not experience abuse, a parent with emotion dysregulation can have difficulties teaching their child how to regulate their own emotions. Emotion regulation is a skill that must be taught to children. If a parent cannot model effective emotion regulation techniques, the child may develop emotion dysregulation. In conclusion, physical, sexual, and emotional child abuse are associated with emotion dysregulation. Children who face abuse can face emotion regulation difficulties at the same time. Not only that, adults who encountered abuse when they were younger faced emotion regulation difficulties many years later. Intergenerational effects are also a possibility. In order for children who encountered abuse to have healthy levels of emotion regulation, early intervention is crucial. So next steps should be taken to find ideal points of intervention. And lastly, I would like to thank my mentor, Emma, for helping me with this project. She taught me how to navigate through different research papers and write a review paper, and I wouldn't have been able to finish my project without her help. And these are some of the references I used for my review paper, and here are the links to the pictures I used in my presentation. Thank you for listening to my presentation.
Awesome. That was wonderful. Do we have any questions from the audience? Anybody? Well, I have a couple of questions for you. So one of them is, so what was the origin of where your project idea came from?
I think just as I gained an interest in child psychology, I thought about what are the most important problems that are present in the world today. And one thing that came up while talking to my mentor was child abuse. And then so from there on, we decided that we could study the effects of child abuse on emotion regulation.
Awesome. Yeah, no, that's, I mean, it's interesting how the ideas kind of just develop from, one after the other like that. That's really cool. Well, great. If there aren't any other questions, we'll actually have Katie go next, it looks like, now that she has jumped into the room. So thank you again. Really appreciate it. Great job. And would you mind either A, clicking the leave button up at the top right, or I can also just do that for you.
I can click the leave button.
Wonderful. Thanks. So I'm going to go ahead and invite Katie up. I probably forgot to do this at the beginning, Claire. I'm really sorry about that. But so Katie is on the left 11th grade student, I believe, from Bernard's High School in Bernardsville, New Jersey, it looks like. Awesome.
I'm 12th grade student.
Oh, 12th. Okay. Yeah. Looks
Yeah.
like my information is a little outdated. So just as a reminder, I'm going to basically allow you to present at about 6 minutes in of the total of 8 minutes. I will add my video back just as kind of a 2-minute warning, and I'll post something in the chat as well for you. Okay.
Thank you so much.
Awesome.
Okay. So starting off, the title of my presentation here is Memory Acquisition, a Study of its Present and a Look into Future Technologies. And let's see. So this is just a little slide about me. I'm a senior, I'm 17 from New Jersey. And my project for Polygence was a literature review on fear memory acquisition and including points on synaptic plasticity and optogenetic research. And I want to give a shout out to my mentor, Sal, who helped me for the past several months perfecting this and really helping me grow in my knowledge of neuroscience. Continuing on. Okay, so I just want to start off basically with what is memory? And this may seem a bit of a rudimentary topic, but I think it's really important. So I want to ask everyone, what does their childhood bedroom look like? Or what does a lemon taste like? What does the alarm that wakes you up every morning sound like? So most likely an instantaneous image or sound will pop into your head without much effort when you recall these things. Where do these images come from? And how are we able to see a room or taste a lemon without currently experiencing these phenomena? We are able to recall them because of our brain's capacity for creating and recalling memories. Many people associate a good memory with tests and trivia games, but memory itself is much more pervasive and important in a person's daily life, governing almost everything they do. However, despite its very real influence, memory is such a broad concept that seems difficult to quantify. Key insights in the field of neuroscience, however, reveal that memory is not as intangible and enigmatic as once thought. For starters, we know that memory is partitioned into 3 specific groups: acquisition, consolidation, and retrieval. So for the purposes of this presentation and my project, we will study acquisition within the context of fear memories, which is the most well-researched type of memory. And we will discuss the brain regions involved in memory, as well as the molecular processes specific to acquisition. Okay, so how is memory acquisition studied? So the process of acquisition is the first and perhaps most recognizable facet of memory, where the memory itself is formed and learned. It has been extensively studied in labs using animal mouse models due to the generalization of their behavior to humans. So fear itself in a mouse model is specifically manifested in observable behaviors such as freezing, and it allows researchers to know if a particular associative fear memory has been formed. So the acquisition of memories is often studied through a process of Pavlovian or classical fear conditioning in mice, and consists of pairing an unconditioned aversive stimulus, referred to as a US, such as a foot shock, and a neutral conditioned stimulus, notated as CS, such as a tone that when presented slightly before the US, leads subjects to develop an association between the 2 external cues. So through experiments and research over the decades, neuroscientists have concluded that a major brain region found to be responsible for the acquisition of memory is the amygdala, a small collection of various nuclei located in the brain's limbic system. When a memory is created, environmental stimuli enter the brain as electrical signals, traveling to the amygdala from other brain regions. And if each signal fires and enters the amygdala within a close enough interval, a memory begins to form. So kind of closing the lens here, we're going to talk about the molecular process of memory. So memory can be broken down even further. And scientists have observed that permanent changes occur in the structure and makeup of many of the neurons in the lateral amygdala, a subpart of the amygdala, where signals converge after mice undergo Pavlovian or classical fear conditioning. If the conditioned stimulus and unconditioned stimulus signals repeatedly activate one neuron and flow through its synapse to activate an adjacent neuron in quick succession, the strength of the connection between the 2 neurons will increase. Persistent stimulation of these neurons and the connection between them has been proven to alter their physical structure, leading to increased postsynaptic receptors and spine density in these neurons. This proves that memory itself exists within the brain and manifests itself in observable changes to the structure and function of the brain. This is so important because it proves that memory is governed by distinct perceptible biological processes, and this gives scientists knowledge about how to study memory in ways previously unimagined. Moving on, I'm going to talk about engrams. So one persistent question scientists have faced is whether memory can be deconstructed even further into the smaller substrates besides just brain regions. When fear memories are acquired, scientists have found that this process spans across an interconnected web of neurons within the amygdala and across other brain regions. This collected ensemble of neurons, dubbed an engram, has been found to be a biological representation of a memory itself. A memory, once thought to be undefinable in its own right, is now known to be stored in these engrams, which can be studied and manipulated in order to ascertain relevant information about memory acquisition and the study and nature of memories themselves. Studies have proved the existence of engrams. By artificially turning off or on the cells that make up an engram, a fear memory can be unlocked or suppressed, showing that engrams are the epicenters of these memories. These studies highly suggest that engrams themselves are the key to understanding memories, as simply manipulating engrams themselves without any external input is enough to block or arouse learned fear behavior. Additionally, engrams have been found, silent engrams, which I have referred to below, are engrams that are found to be still intact, but their connection to other brain regions, such as the thalamus, which is the relay center of the brain, has been severely weakened, and this will be important as we move forward. So what is really important is the implications of this knowledge as a whole. So because we now know that memory can be manipulated fundamentally in a biological way, and we don't have to think about memory as such an ambiguous topic, neuroscientists have been able to create and test novel technologies using knowledge regarding memory acquisition and the understanding of the biological constructs of memory. The use of optogenetics, an invasive technology that uses light to control neurons, has opened up various new possibilities in terms of treating memory conditions and even manipulating memory itself. For instance, a study performed in the labs of MIT in 2013 found that they could implant fake fear memories in mice using optogenetics. So this entire study of memory acquisition is important because memory manipulation is becoming an increasingly real possibility despite its sci-fi undertones. One possible use for optogenetics that is pertinent to the acquisition of memories is to combat or prevent PTSD and traumatic memories. Optogenetics provides hope to those at risk for developing traumatic memories, such as soldiers during wartime. It is possible to inhibit neurons that are significant in acquiring new memories instead of retroactively attempting to silence traumatic memories that have already formed. Additionally, artificial stimulation of these silent engrams referenced before have been found to bring back forgotten memories in mice, pointing to a potential treatment for some neurodegenerative diseases, as with a possibility of memory manipulation. However, comes a whole host of other unanswered questions, such as whether or not these methods should be used at all, and if so, in what context. However, if the necessary questions are addressed properly, these new technologies will take steps to help people in ways we cannot yet imagine. Thank you. And you could contact me, my email's there. You can also find me on LinkedIn. And here are some of the references I used for my review and for the presentation. Thank you so much.
Well, that was wonderful. Do we have any questions? If you guys want to post these in the session chat, feel free. Otherwise, I have a few questions that we can come to in a minute or so. Any questions? If you'd like to ask your question, feel free to just say, something in the chat so I know who to add, because a lot of people are trying to share their screen and I'm not really sure about who is actually wanting to ask a question. Okay, well, maybe I'll just ask my questions if you don't mind. So in terms of the structure of like fear conditioning, so what was the kind of, how did you end up with that? Like, where did that come from? Like, what was the interest in that originally?
Yeah, so the impetus for my project as well came from my limited knowledge of neuroscience going into Polygence. Specifically, I had proposed an idea to look at something regarding synaptic plasticity, which I talked about is kind of the change in the structure of the brain over time with new memories and learning new things. Right. And I thought that was the coolest thing ever. So with my mentor, Sal, we just kind of fine-tuned that a little bit and used some of his expertise. And he introduced me to optogenetic research as well,
Yeah.
which was something that I had not seen before and I thought was very, very cool. So we did look at that within the context of fear memories, because fear is very easy to study because the behaviors shown in mice models and other animal models are very easy to see. Like mice usually, they freeze in one spot when they feel fear. So it's easy to see and manipulate those behaviors.
That's really cool. And so I have one final question. Actually, looks like there's a question here. Can you say something about the contamination of memories, or the fact that memories can be corrupted?
Yes. So without getting into it in the presentation, there is a whole host of unanswered questions about implanting new memories, forgetting memories, and certainly, yes, memories can be corrupted. There are, limited knowledge about the actual applications of this in humans so far. Yes, definitely, I would say 1984-ish. It's kind of crazy because people who can administer these sort of treatments and optogenetic research, it's very important that they aren't corrupted by this power as well. And so whatever they do, the ethical considerations are at the forefront of this kind of discussion, because it gets into a lot of ethical issues, even legal issues. And so there probably needs to be specific boundaries set before anything like this would be able to happen. And even then, you can't really guarantee that these technologies will be applied equally, will be applied fairly, and will be applied in a way that doesn't hurt people.
Yeah, that's great. Well, thank you very much. I really appreciate that talk. It was wonderful.
Thank you so much.
So if you wouldn't mind either leaving via the button up at the top right, or I can also help you exit.
Of course. Thank you so much. Bye.
So up next, we have Angela Predolac. I hope I said that correctly. If I didn't, feel free to correct me in just a second here. They are a 10th grade student, or maybe 11th grade if my data is a year old now, in Hunter College High School from New York City, New York. And so thank you very much for being here. Oh, we met before. Awesome. So I'll go ahead and let you share your screen and go on with your presentation. And just as a reminder, when you see my video come back, this will basically be the 2-minute warning. Okay?
Okay. Can you see my screen?
Yep, can see you.
Okay. Hi, my name is Angela, and my Polygence project is titled A Predictive Model of Pediatric Epilepsy from Visually Guided Saccades. So before I start, I just want to share a little bit about me. I live in New York City, but both of my parents are originally from Serbia, and some of my favorite hobbies include Zumba, traveling, and reading. I attend Hunter College High School, and I just started my junior year. And some of my school involvements include being a member of the Girls Who Code Club and a mentor for our writing center. And my project is a combination of computer science with epilepsy, because I've had a long time interest in computer science, math, and problem solving, and I have a cousin who was diagnosed with epilepsy as a baby. So I came into Polygence wanting to learn more about these two things. I'll start with an introduction to epilepsy and its diagnosis. Epilepsy is a neurological disorder categorized by seizures and abnormal brain activity. Left untreated, it can lead to many complications, including brain damage, reduced quality of life, and sometimes even death. So early and accurate diagnosis is really important. In my project, I reference two main subtypes of epilepsy, which are chronic or treatment resistant, and controlled, which is treatment responsive. Typical diagnosis of epilepsy includes consultation with the neurologist, who performs a few brain scans, the principal one being an electroencephalogram or an EEG. And as you can see in this image, electrodes are strapped to the brain in order to record the epileptiform activity. But there's a few problems with this. Firstly, diagnostic EEGs fail to correctly identify epilepsy in about half of patients. Brain scans can also cost hundreds of dollars, and epilepsy rates are even higher among socioeconomically disadvantaged communities. Finally, as you can see in the image, having electrodes strapped right to your head can cause a lot of discomfort and anxiety, especially for young children, and especially because neuropsychiatric disorders are highly comorbid with epilepsy. So because of this, I wanted to explore a low-cost and non-invasive way of diagnosing epilepsy. So I turned to machine learning based on eye movement data. Saccades are a type of rapid eye movement that abruptly change the point of fixation. So an example is if you're driving the car and then you quickly and accurately look towards a stop sign close in front of you. Previous studies have observed saccadic eye movement abnormalities in children with epilepsy, and saccades have also been used in machine learning predictors for autism, Alzheimer's, and Parkinson's, all of which are neurological disorders. Based on this, I hypothesized that saccadic eye movement data could be used to predict pediatric epilepsy and subtype. I was able to find an open source dataset measuring the saccadic eye movements of healthy children and children with chronic or controlled epilepsy. The cohort consisted of 74 kids, 11 with chronic epilepsy, 15 with controlled, and 48 who were part of an age and sex matched, typically developing population. 74 is a sample size not large enough to train effective machine learning models, but the study conducted 30 pro-saccade trials and 30 anti-saccade trials for each subject. A pro-saccade is when you look towards a new target, and an anti-saccade is when you look away from it. So in this diagram, you can see that the patients looked at a centrally fixated cross for 1 second, and then a green circular target appeared to the right or left. And if it was a pro-saccade task, they had to look quickly and accurately towards the new target. And if it was an anti-saccade, they had to inhibit a pro-saccade towards it and instead look to the opposite side of the screen. So I decided to use each trial as an observational unit for model training, which gave me over 4,000 examples to train the models on. And after data was cleaned and pre-processed, I ended up with 6 predictor variables, all of which were saccadic eye movement parameters, including accuracy and other dynamics. I had 6 candidate model architectures or algorithms for classifying that I trained using 70% of the data and tested using 30%, which is called holdout validation. I selected the best model according to 2 measures of evaluation. Accuracy and AUC, or area under the receiver operating characteristic curve. If you plot the false positive rate on the x-axis and the true positive rate on the y-axis at various threshold levels for a machine learning classifier, the area under that curve will represent how good the machine learning model is at identifying the correct classes. So an AUC value of 0 would mean that all the positive classes are being labeled as negative and vice versa, while an AUC value of 1 would mean that all the examples are being put into the correct class. I then calculated the predictor importance estimates, which tell me how much weight the model is assigning to each of the variables. Finally, I performed significance testing to show that the model is significantly above chance. For epilepsy diagnosis, the best model architecture was an ensemble classifier, which achieved an AUC of 0.66 and an accuracy of 67%. As you can see in this image, all the models were well above chance, which is represented by the dashed line, and they all achieved pretty similar performance. For the predictor importance estimates, peak saccade velocity, representing the speed of the saccade, had the highest importance, with saccade amplitude, or distance traveled by the eye, and start time, or time taken to initiate it, shortly behind. And 5 out of 6 of the variables had some importance. For the subtype model, the best architecture was also an ensemble, which achieved an AUC of 0.75 and an accuracy of 72%, outperforming the diagnostic model. Once again, all the models were above chance, but this time the ensemble stands out a little bit more as having the greatest area underneath. For epilepsy subtype, this time there were 2 methods of calculating the predictor importance because it was a slightly different type of ensemble. But the saccade amplitude, velocity, start time, and duration of the next fixation all had the highest importances, with all the variables having some importance. This suggests that the optimal model for predicting epilepsy and subtype based on eye movement data is an ensemble classifier, and both the models achieved significantly above chance performance. They achieved robust AUCs and high specificities, which means they were good at detecting the negative cases, which would be controlled epilepsy or no epilepsy, but they had lower sensitivities, meaning it was harder to identify the positive cases, which would be epilepsy or chronic epilepsy. This may be because the data set was skewed to include more negative examples, so the models were better trained to identify those. The epilepsy subtype model outperformed the diagnostic model, which may suggest that differences in eye movements between controlled and chronic epilepsy are even more pronounced than those between healthy control and epilepsy of any subtype. but it might also be because the data for the subtype model was a little bit more balanced in terms of class distribution. 100% of the children with epilepsy were correctly identified for at least one trial, which suggests that they present with saccadic abnormalities only some of the time and not ubiquitously. Performing multiple trials could lead to a higher diagnostic accuracy in practice. According to the predictor importance estimates, saccadic parameters like speed and time taken to initiate it are heavily influenced by epilepsy. And all of this suggests that the model can be used as a low-cost and non-invasive supplement to an EEG. And here you can see a little girl using the eye tracking data, and it's so much less invasive than the image of the EEG shown at the beginning. Future directions include external validation or testing this model with a different population, as well as prospective studies or collecting new data to test the model on. A larger cohort size would improve the generalizability of the results. As you can see in this figure, epilepsy also has a lot of comorbidities, including ADHD, depression, anxiety, and more. So a dataset testing the model with non-typically developing control groups would help isolate epilepsy as the sole cause of the abnormalities. Finally, a dataset measuring the eye movements before children were classified as chronic or controlled and treated would help predict treatment response to anti-epileptic drugs in advance. I'd like to end by thanking my wonderful mentor, Olivia, the whole Polygence team, and my family for all their support in this project. And I'd like to thank you all for listening.
Wonderful. Great job.
Thank you.
Are there any questions? I thought it was very informative.
Thank you.
Okay, well, maybe I'll pose a question while people consider what they might ask. So in terms of the future directions, I'm just kind of wondering, so you've kind of established this modeling outline here. So in terms of, incorporating larger cohorts, like, are there certain things about those cohorts that you would want to kind of include in future iterations of this?
Yeah, I think that definitely a more balanced data set in terms of positive to negative examples might lead to higher accuracy because the models were better at detecting the negative cases because the data set that I used had more of those to train on. And also what I mentioned here, having like controls that people who don't have epilepsy but have like ADHD or depression, anxiety, and some of these would help show that epilepsy is causing the saccadic eye movement abnormalities and not a comorbidity. So those are some things that I would want to include.
Gotcha. So I see that there's a question in the Q&A asking how long you were working on this project.
So I was working on this project over the summer. So I'd say about three to 4 months.
Wow. Well, does anybody else have any other last minute questions they want to sneak in here? No? Okay. Well, awesome. Well, thank you again, Angela. That was wonderful. I really enjoyed that. So either clicking the leave in the top right, we'll just go ahead and remove that. So why don't we, in the meantime, we'll transition on to Sanjana. Sanjana. And so I'm going to go ahead and add you to the screen. And we'll have you go ahead and start your presentation. And hopefully, if Nisha is able to get back, we'll resume from there. Awesome. So feel free to go ahead and share your presentation. And then once you see my video again, that'll be the 2-minute warning. Okay.
Oh, never mind. Sorry, I figured it out.
Just as a reminder to everybody, due to
Can you
the fact that we're on an online option, if you can minimize other tabs that you might be on, it'll also help to ensure that there's no instability in the connection. Okay. All right. I'm going to leave the video, and when you see my video come back, 2 more minutes left. Okay.
Okay, can you see my presentation? Hi everyone, my name is Sanjana Srimath, and my Polygence research project is about CDKN1C hypermethylation patterns in human breast cancer. So a little bit about me. I'm a senior from Ellicott City, Maryland. In my free time, I really enjoy reading, especially historical fiction and mystery novels. Other interests of mine include tennis and playing the veena, which is an Indian classical instrument. I would like to really thank my mentor, Ms. Taylor, for guiding me throughout the entire research process. So I enrolled in Polygence having a general idea that I wanted to do a research project involving bioinformatics and oncology. With the help of Ms. Taylor, I decided to focus my research on CDKN1C, which is also known as P57 or KIP2, which is a tumor suppressor gene from the CIP/KIP protein family. Its main function is to regulate cell proliferation or cell growth by inhibiting the cyclin D1 and CDK4 complex during the G1 phase of the cell cycle. And you can see that represented on this image. I do want to mention that this image is a very simplistic overview of the cell cycle, and there's a lot more going on, but I just really wanted to highlight the protein I researched. So the cyclin D1 and CDK4 complex drive cell proliferation at the G1/S phase of the cell cycle, and many proteins regulate this process to ensure that only healthy cells can proceed. CDKN1C being one of those proteins. Therefore, when CDKN1C does not function properly, the cyclin D1/CDK4 complex is unchecked, and that will allow for rapid cell proliferation. In fact, overexpression of cyclin D1 has been reported in upwards of 15 to 25% to even 50% of breast cancers. Additionally, previous studies have shown that CDKN1C expression is low in breast cancers, and the loss of CDKN1C is associated with a lower survival rate. So CDKN1C is located on chromosome 11p15.5, and this is significant because this region has been associated with sporadic cancers and epigenetic abnormalities, such as hypermethylation. I also learned that CDKN1C can be inactivated through loss of imprinting, which is the loss of the normal allele of the gene. And this is also controlled by epigenetics, including DNA methylation. So this further prompted me to really focus on the role of hypermethylation. And DNA methylation is an epigenetic mechanism where a methyl group is added to the DNA strand. So based on previous research, the hypermethylation or overmethylation of CpG islands, which are the sites where DNA methylation occurs, on CDKN1C has been associated with a loss of gene expression, and this directly impacts the progression of several cancers. So this study utilizes computational tools to analyze the extent of CDKN1C hypermethylation in different stages of breast cancer. I used Ensemble, a genome database, to analyze the CDKN1C gene and its surroundings, and I found that there were many CpG islands on this gene. The OE was equal to 0.89, and this represents the density of CpG islands. This was a crucial piece of information because previous research has found that CpG islands, which are typically unmethylated or have really low methylation levels, are correlated with a loss of gene expression and can really contribute to the silencing of tumor suppressor genes such as CDKN1C when hypermethylated. The Ki67 expression, which was also a marker of cell division and growth rate of breast cancer, was shown to be higher in advanced stages of breast cancer. So this means that as the tumor stage is higher, it can be expected that cells are dividing at a faster rate because they're not being controlled by tumor suppressor genes. Based on the background information I learned from previous research papers, my hypothesis was that hypermethylation at the CDKN1C locus would be positively correlated with the more advanced tumor stage, because if hypermethylation on the CDKN1C gene causes gene imprinting and loss of gene expression, then the CDKN1C protein would not be present to regulate the cell cycle, and this would lead to the proliferation of cancerous cells. I began by loading all of the DNA methylation files from the TCGA/BRCA dataset. And this is a database that's provided by the National Cancer Institute that has thousands of breast cancer files with clinical and patient information. So these files were loaded on RStudio software, and specifically I focused on analyzing the 342 files from the Illumina Human Methylation 27 platform. And this is a specific probe that measures methylation levels in a sample of DNA. Next, I created a data frame that compiled patient information, such as tumor stage, vital status, and age, with methylation beta values from CPG islands in the CDKN1C locus. And these beta values were mined from each of the individual 342 methylation files. Beta values represent the intensity of methylation at a particular CPG island, and they range from 0 to 1. 7 CPG islands in the dataset map to the CDKN1C gene. I use box plots to visualize the methylation beta values at each of the 7 CDKN1C CPG islands as grouped by tumor stage. So the CPG islands in the first row appear to be more highly methylated than the other 5 CPG islands. The second row of CPG islands have high variation in extent of methylation in some specific tumor stages, for example, in tumor stage 2A and 2B. Notably, the third row of CPG islands have extremely low beta values, and they showed distinctly higher methylation levels, specifically at stage 1B. And I think this can be explained by a very low number of patients categorized as stage 1B. And then the last CPG island has low methylation levels for all tumor stages, noting that y-axis scale. So this suggests that methylation may have a more notable effect on some tumor stages compared to others. The first CPG island, as you can see in this image, shows a trend of increasing beta values with more advanced tumor stage, but I ran a chi-square test on RStudio, and this trend was found not to be significant because the p-value equaled 0.4487. Thus, there was a lack of convincing evidence to support the hypothesis that CDKN1C methylation increases with breast cancer tumor stage. To relate this finding back to gene expression, I created a plot depicting CDKN1C gene expression by tumor stage, but there was no clear trend in gene expression levels between the different tumor stages. Additionally, I created a circle correlation plot to show correspondence between extent of methylation at different CPG islands. So as seen in the plot, some pairs of CPG islands have high positive correlation, while others have near zero correlation. The darker the blue circles are, the more positively correlated the CPG islands are. So the main takeaways, overall, while the hypothesis was not supported by the data analysis, methylation at some CPG islands might be coordinately regulated, and methylation at other CPG islands on the CDKN1C gene might reflect entirely different regulatory mechanisms. But definitely more research is needed to understand the methylation patterns and the relation to the complex changes that occur throughout breast cancer development and progression. So here are the references I used for this presentation and my research paper. Thank you so much for listening to this presentation. I would like to acknowledge the Polygence team for giving me this amazing opportunity to conduct this compelling research over the summer. And again, many thanks to my mentor, Ms. Taylor. Thank you.
Awesome. Well, are there any questions from the chat? Anybody have any questions they'd like to pose? Well, I had a couple of questions for you. So in terms of the evaluation of the biochemistry, right, that's associated with the epigenetic changes, how did you find the learning process for that? Like, how hard was that to kind of digest at the beginning?
I would say it was hard. I started with learning about generally like the cell cycle, and then I came across the cyclin D1 and CDK4 complex, which I wanted to really focus on, and what factors affect that. And I generally had to do a lot of research because there's so many proteins, involved in that part of the cell cycle. So eventually, I focused on CDKN1C in particular because I think there was a little less research on this particular protein.
Right.
So yeah, it definitely, I had to read a lot of research papers and, kind of focus on this protein.
So we have a question in the chat of how long were you working on your project?
I think about 3 months.
Yeah. Oh, wow. That's awesome. So I have one final question for you. It's that, so the CpG island, right, in this phosphorylation of these areas is a wide disease problem. Like, this is something that's associated with a lot of other diseases. So I was kind of curious, like, when you were going through the reading for, the breast cancer associated genes and all of this, did you find other genes that you found interesting that you might try to explore later on?
Yeah, so that entire, the region 11p15.5 chromosome, there are a lot of proteins on there, or genes on there, sorry. I don't remember the name of the genes off the top of my head, but I did notice several genes near the CDKN1C locus. And while I was reading some of the research papers, there were some mentions of those proteins that were, they affect other cancers and, have been implicated in having epigenetic abnormalities. So definitely, I think it would be interesting to research that entire region in the future.
So we have a question about the flowchart diagram in your presentation, and someone wanted to just take another look at it. And so just so everybody knows, the session will officially be over as of now, technically. But I did want everybody to know that there's a couple of minutes before the next series of sessions start. So just feel free to hang out.
So I believe this is the flowchart. This is basically my hypothesis.
This is awesome. Well, very cool. Are there any other questions? No? Okay, well, then this concludes the session 7. And I'm sure I'll see some of you at the upcoming sessions. Hopefully you guys will go and check out some of the other talks that are happening throughout the day. And thanks again, everybody, for joining.